If you recall from last chapter, we said that if you look at the karyotype, there are 46 chromosomes arranged in 23 pairs, and one pair of those chromosomes are the sex chromosomes, and the other 22 pairs are called autosomes. So in our last screencast, we looked at sex linkage, that is traits that are located on the sex chromosomes. And so in this screencast, we're going to look at autosomal linkage and some things that can happen with those with those other ones. So let's take a look here. And we're going to start with a problem, and it's a dihybrid cross involving fruit flies. Don't be troubled too much by the symbols. Basically, B is coding for body color, so gray or black. The wild type is gray. That's what most fruit flies are, but then there's this variety of black colored. And then VG refers to the wings, where the wild type wings, normal wings, are VG+, and then what are called vestigial wings, or these kind of non-functional wings, are VG. So if we have a fly that is heterozygous for body color and wing type, crossed with a homozygous recessive fly, what happens? Well, let's do the Punnett square and find out. So here we have our cross, and just like before, we got to make sure, based on the law of independent assortment, that we have each B allele combined with each type of VG allele. So B plus, VG plus, B plus, VG, B, VG plus, and B, VG. And then over here, basically the only gamete this organism could produce would be a B, VG, and I could write it 4 more times, but I think you'll see that if I do that, it's just going to end up being the same thing 4 times. So basically we have a 1 to 1 to 1 to 1 ratio, or a 4 to 4 to 4 to 4 ratio. So we're just going to stop. Now if you fill that in, which I'm confident at this point you could all do, let's see what results we expect. And there it is. We would expect a 1 to 1 to 1 to 1 ratio of B plus VG plus, B VG plus, B plus VG, and B VG. And which of Mendel's laws cause you to predict this? The law of independent assortment, right? Just like we've said. So let's actually go to the lab and do the experiment and see what we get. Turns out we get 42% this genotype, 41% not. Huh? What the heck's going on? So we predicted this based on Mendel's laws, but we got something drastically different from that when this experiment is actually carried out. So clearly there's something going on that Mendel did not understand. So let's try and figure out what is that. The thing that it is something called autosomal linkage. Autosomal linkage occurs when 2 genes are located together on the same chromosome. If they're on the same chromosome, they tend to be inherited together. And so to understand this, let's just kind of take a look here. So if we start as we did with this heterozygous individual, we could draw out these chromosomes. And it turns out that the alleles for B plus and VG plus are located on the same chromosome like this. And so as a result, they are mostly going to be passed on together. The B plus, VG plus will kind of, if you could imagine this lined up at meiosis, you know, one is going to go this way, one is going to go that way. We can do it horizontally, I suppose. But either way, these 2 chromosomes are going to separate out, and most of the gametes are either going to get B plus, VG plus, or B, VG. And those are the parental phenotypes, meaning these offspring have sort of inherited the chromosome as it was arranged in the parents. Now, it is also possible, though, to get what are called recombinant phenotypes. These result from crossing over. So in our last chapter, we talked about how during prophase 1 of meiosis, crossing over occurs where B plus and VG plus can like swap little sections. And so if we do that, then all of a sudden the B plus is going to be combined with VG, and the B is going to be combined with VG plus. So now we have a B plus VG and a B VG plus. These are called the recombinant phenotypes, and they're less frequent because it depends on this crossing over event to happen between them. So let's pause and just kind of model that to make sure we understand what's going on. So here's a picture that we drew in the last chapter about meiosis, and we basically said if you started with big A, big Y, and a, little y, crossing over can happen to create this new unique combination of chromosomes. So it's just exactly what we're talking about. It's the same thing. So I could just replace, the A with a B plus and the Y with a VG plus, and basically it's the exact same phenomenon here. Okay, so in this case, the big, these two chromosomes would be the parental if they were passed on intact, but then if crossing over occurs, it results in these recombinant combinations. If you don't like that, I got another way to explain it. Our Skittles, remember our candy bar? Let's zoom out a little bit here. So here are our chromosomes, and you can see that the B and VG alleles are on the same chromosome. They're both on the Skittles. So here we have our parental chromosomes, B plus VG plus, and B and VG. And so what happens is, during meiosis, during interphase, the chromosomes replicate. In prophase 1, they kind of pair up together, that process called synapsis, where they sort of join together, and then crossing over occurs. And when they cross over, then you get these new combinations like that. So if this crossover event does not happen in this particular location, then during meiosis, these are going to separate out into opposite ends of the cell, and half your gametes are going to be B plus VG plus, while half are going to be B VG. That's the parental chromosomes. If, however, the crossover event does occur, then when they separate during meiosis, now it is these recombinant phenotypes that are going to arise, B VG plus, B plus VG. So it all depends on if that random crossover event happens to occur between or at this particular location. Yeah? So the B, VG tend to be inherited together, but the crossing over can mix it up. So once we have these results, we can actually use this to do something kind of interesting here. We can actually use this to get a sense of where these different alleles are located with respect to one another on a chromosome. So to do that, the first thing we need to do is calculate what is called the recombination frequency. That is the percentage of recombination. And it's a very simple calculation. You simply take the number of recombinant gametes divided by the total number of meioses. So what that looks like, if we were to switch to this piece of paper, is, so the number of recombinants, I forgot my numbers, is going to be these recombinants. So 206 plus 185. And so switching back, 206 plus 185, because those were my recombinant phenotypes. And then we're going to divide that by the total number of meioses, which is 965 plus 944 plus 206 plus 185. And if you do that calculation, you get an answer. And I don't know what that answer is. Hold on, let me pause. Okay, take two. And the answer is, oh, there it is, 0.17. Now these are often expressed as a percent. Basically, we just calculated that there is 17% recombination. So these two recombinants divided by the total are about 17%. So what is the value of that? Well, the higher the recombination frequency, the further apart two alleles are on the chromosome. And this should kind of make sense to you because crossing over, it happens randomly along the length of the chromosome. And so the further apart two alleles are, the more likely that crossover event will happen between them. Here's a visual that kind of explains that. You've got the A allele and the B allele spaced really far apart. And so any position here, if crossing over just happens to occur in any of these spots, that could potentially separate this big A and big B from one another. Versus on this diagram, here they're really close together. And so really the only place where recombination can happen is exactly right in this spot. And so it's much less likely to happen between these two genes, so you would expect a lower recombination frequency. So the recombination frequency gives you a sense of how far apart two genes are on a chromosome, and that allows us to make these sort of preliminary genetic maps. And so scientists sort of created the convention that 1% recombination frequency would be equal to 1 map unit. And so, and then if genes are on separate chromosomes, their recombination frequency is going to be 50%. And if that doesn't make sense to you, let me explain using the dot cam. Basically, we can see that here, where it's basically the law of independent assortment, right? So if they're on separate chromosomes, as the A and B alleles are here, basically they can line up this way during meiosis, in which case big A will be with big B, or they can line up this way during meiosis, in which case big A will be with little b. And so basically the recombination frequency is 50% because big A is with little b 50% of the time, and big A is with big B 50% of the time. So a 50% recombination frequency could mean one of two things. It could mean that the genes are on separate chromosomes, or it could mean that they're super far apart on the same chromosome to the point where there's pretty much going to be a crossover event no matter what between them. And it turns out that some of the traits Mendel analyzed are actually on the same chromosome. And again, they're just so far apart that it looked like they were on different chromosomes, and so he kind of lucked out and was able to develop his laws despite this linkage business. So then, like I said, this allows us to make a preliminary genetic map. So let's assume that you've learned that, A and B have a recombination frequency of 13.2%, B and C 6.4, and A and C 18.5. So we know that these are all on the same chromosome, and so what we do is we say, all right, well, A and C must be the furthest apart because they have the largest recombination frequency between them. And so then it must be, A and then B must be in the middle. And so basically A and C are 18.5 map units apart. A and B are 13.2, and B and C are 6.4. Now the astute mathematician among you might say, hey, wait a minute, 13.2 plus 6.4, that's 19.6. That's not 18.5. Why isn't it perfect? And the reason why it's not perfect is because of that element of random chance. So if you were to do these crosses a bajillion times, then yeah, it would probably add up to pretty close to exactly the right number. But in the absence of that, random chance is going to make recombination happen a little bit more or a little bit less than would be predicted under perfect circumstances. And so there is a little bit of wiggle room there when these numbers add up. So if it's close, then that's probably a sense that you're on the right track with this mapping business. So basically, so if the chromosomes are on different genes, then Mendel's law of independent assortment kicks in. If they're on different chromosomes. If they're on the same chromosome, then they can be linked and they can travel together, and this linkage idea kicks in. That's it. Thanks, everybody.